Using the BioModelAnalyzer to model biological networks. Proteins are essential to mammalian life. They assist with DNA replication, catalyse metabolic reactions, and respond to stimuli which are essential for cell homeostasis. However, they very rarely act alone. Protein-protein interactions are a necessary part of the cell's life cycle. This requires constant communication between proteins with differing and specific functions, and pathways where proteins are communicating with each other in order to produce consequences that are beneficial to cell changes. In addition, a crucial part of development in carbon-based life forms requires sophisticated interactions amongst biological networks within cells as well as between cells in order for some cells to develop while others remain undifferentiated. Mammalian somatic cells house identical copies of DNA, but gene expression will need to be varied between them in order for some cells to become specialized. In some cases, cells will need to communicate with their neighbours in order to regulate the level of their own specialization. An example of where this occurs is in self-regenerating tissue, where cells can differentiate in order to form several layers beneath the surface of the skin. Layers of differentiated skin cells are able to form because of crosstalk between two protein signalling pathways, the Notch and Wnt signalling pathways. The Notch and Wnt pathways are essential to the regulation of cell proliferation, and differentiation and abnormalities in the functioning of these pathways are linked to several types of cancer. The Wnt signalling pathway gets its name from Wnt, a short-range signalling molecule that binds to receptors of the frizzled family. Wnt signalling results in the degradation of a protein called beta-catenin through a series of steps involving other proteins. Binding of the Wnt signalling molecule to the frizzled receptor leads to the phosphorylation of a protein called dishevelled, and dishevelled inhibits the action of axin. Axin then marks beta-catenin for degradation. The inhibition of axin allows beta-catenin to accumulate and travel to the nucleus, where it activates DNA binding proteins. Wnt signalling is therefore responsible for activating certain target genes. Notch signalling is named after the Notch receptor protein. When immediately neighbouring cells express ligands, for example jagged, on their surface, the Notch protein is cleaved and the intracellular domain of the receptor, Notch-IC, travels to the nucleus. This leads to the activation of the transcription of the target genes of Notch signalling. One of the targets of Notch signalling is P21, and P21 is a negative transcription regulator of Wnt. If we use the single variable GT1 to represent all the downstream targets of increased levels of beta-catenin, and GT2 to represent all downstream targets of Notch-IC, we can show the crosstalk between the two pathways using this simple diagram. Jagged is activated by GT2, and this creates a positive feedback loop in the Notch signalling pathway. This Notch-Wnt pathway model can be scaled up to simulate the skin cells in the epidermis, keratinocytes. 5 identical keratinocytes labelled cell 1 to cell 5, representing the mammalian epidermis, can be used to mimic a different layer of skin cells. The left side of the model represents lower layers of the skin and the right side represents higher layers. The leftmost cell is part of the basal layer where the level of Notch receptor is lower than in the suprabasal layer. Reduction of Notch activity in the bottom layers makes sure that the cells remain undifferentiated, whilst as you move up the layers, increasing Notch activity and decreasing Wnt activity promotes correct differentiation. Notch can therefore be set to off in cell 1, to medium in cell 2, and to high in cells 3 to 5. Cell-to-cell communication can therefore be modelled by connecting signalling molecules emulating Wnt and neighbouring cell ligands to Wnt and jagged respectively of the immediately neighbouring cells. A pathway like this can be modelled using the BioModelAnalyzer, and in fact, using the BioModelAnalyzer enabled both computational biologists and laboratory biologists to independently come to an identical conclusion, indicating that an interaction between GT1 and jagged was necessary for the two models to communicate, and for the model to achieve stability. For example, when a 5-cell keratinocyte model like that described is tested for stability using the BioModelAnalyzer, the instability of the system is indicated. Once an interaction between GT1 and jagged is added to this model, the tool shows the system achieving stability. If a model is identified as being unstable, then the BioModelAnalyzer tool allows the user to check simulation tables, plots, and carry out further testing, which provides the user with a more detailed analysis of where instability is found, so corrections can be made and a new hypothesis can be suggested. When using the BioModelAnalyzer tool to design a model, already established interactions between proteins from prior experimental data are used. Positive feedback loops, negative feedback loops, protein activation, protein inhibition, and variable protein signaling molecule and ligand concentration are all factors which can be simulated. In addition, models can be expanded to include many cells housing differing pathways and models incorporating communication between cells. In a similar fashion, the BioModelAnalyzer can be used to answer key questions such as, what will happen if I change the concentration of this signaling molecule? How will the system respond if I decide to target a drug to this particular protein? What are the consequences if this protein is mutated and there is a complete loss of function? Variables within the model can be changed and interactions between components can be disabled. The BioModelAnalyzer enables sophisticated models to be built in order to simulate the essential processes involved in cell differentiation. Models developed in the BioModelAnalyzer allow experimental biologists to understand biological systems and drives discovery.